Okay, let's unpack this. Think for a second about how your body manages to read and use the, incredibly complex instructions in your genes, making sure everything runs smoothly. It's happening constantly, right? Without you even thinking about it.
Right.
But what if there was just a tiny glitch in that reading process?
Hmm.
Here's where it gets really interesting. Scientists have recently uncovered a surprising link between some very small molecules within our cells and the development of certain neurological disorders.
Yeah, what's fascinating here is that we're talking about small nuclear RNAs or snRNAs.
Then RNAs.
They're tiny,
Wow.
yes, but their job is absolutely vital. They make sure our genetic information is processed correctly. This new research takes, a deep dive into how even subtle variations in these seemingly minor players can have significant consequences for how the brain develops.
Right. So our genes are like these big instruction manuals for building and running our bodies, but they're not always written in a straightforward way. Imagine a recipe with some extra notes or paragraphs that you need to remove before you can actually follow it. That's kind of what splicing is all about.
Exactly, yeah. Genes contain sections called exons, which are the important parts that code for proteins,
Okay.
and introns, which are like those extra bits that need to be cut out.
Like junk bits almost.
Sort of, yeah. Splicing is the cellular editing process that removes the introns and joins the exons together to create the final usable message, what we call messenger RNA.
Okay.
And that message then tells the cell how to make proteins.
Got it. And these snRNAs you mentioned are essential for this editing process, acting like, I don't know, specialized tools in a cellular editing suite.
Precisely. snRNAs are key components of a larger molecular machine called the spliceosome.
The spliceosome.
Right. Think of the spliceosome as a highly specialized factory assembly line. Different snRNAs act like specific robotic arms on that line, each with a crucial task.
Okay.
Broadly speaking, these tasks fall into a few categories: recognizing where the cuts need to be made,
Mm-hmm.
performing the actual cutting and joining, and ensuring everything is aligned correctly.
So some find the spots, some do the cutting.
Exactly. For example, some snRNAs like U1 are involved in the initial recognition of the splice sites, while others, like U2, U6, form the engine, the catalytic core, that drives the splicing reaction. U5 helps align things. U4 keeps U6 in check until it's needed. It's quite intricate.
Wow. Okay. So the study we're diving into today took a really close look at the genes that provide the instructions for making these snRNAs. They looked in a large group of people with rare developmental disorders. What was the sort of the main thing that jumped out from this research?
Well, the big takeaway, connected this to the bigger picture, is that the study identified new and significant genetic variations in these snRNA genes in many of these individuals.
Oh.
This strongly suggests that these tiny molecules are far more critical for healthy brain development than we perhaps previously fully understood.
Right.
It really highlights how disruptions in these really fundamental cellular processes can be a key factor in complex neurodevelopmental disorders. This is a significant advancement because it points to a previously, underappreciated area of genetic research.
Okay. So let's zoom in on one of these snRNAs you mentioned, U4. The gene that codes for it is called RNU4-2. Is that right?
That's correct, RNU4-2.
And it sounds like this is where the study uncovered something particularly notable.
That's right. The researchers found a significant number of patients with de novo variants in the RNU4-2 gene.
De novo meaning new, not inherited.
Exactly. These genetic changes appeared for the first time in the individual, like a new typo in their genetic code, and weren't inherited from their parents. And what's particularly noteworthy and perhaps a bit unexpected is that many of these variations clustered within a very small, just 18 base pair long critical region of this gene.
Wow, 18 letters. That's tiny. And did the researchers see any connections between where these genetic typos occurred in RNU4-2 and the specific health challenges these patients faced?
Yes, and this is where it gets really interesting and reveals a level of precision we're only just beginning to fully grasp.
Okay.
The study revealed a correlation between the specific location of the RNU4-2 variant within different functional parts of the U4 snRNA molecule
Uh-huh.
and the clinical characteristics observed in the patients. It appears that even very subtle changes in different working parts of this tiny RNA can lead to a whole spectrum of neurodevelopmental features.
So different glitch, different outcome.
Pretty much. This detailed genotype-phenotype correlation for such small molecules is it's a significant step forward in our understanding.
You mentioned a specific genetic change that kept showing up. What was that, and what were some of the common features seen in those individuals?
Absolutely. There was this recurrent insertion, N.6465 NST, Essentially an extra T base inserted at a specific point in the genetic code.
Just one extra letter.
Just one. But it was found in a large number of patients, and it was strongly associated with more pronounced effects. These individuals often shared certain facial characteristics, including a larger mouth, a shorter philtrum, that's the little groove between the nose and upper lip,
Okay.
downturned corners of the mouth, thicker lips, eyes that appeared deep-set, maybe sparse eyebrows, and strabismus, which is a misalignment of the eyes.
Crossed eye sometimes.
Yeah, sometimes called crossed eyes. Interestingly, older individuals with this particular variant sometimes also showed a slight asymmetry in their face.
And there was this really surprising finding about where these RNU4-2 variants came from, wasn't there? It seemed to be more often from the mother's side.
Yes, this was a very intriguing observation, and it raises some important questions for future research. The study found that RNU4-2 variants, especially that N.6465 NST insertion, predominantly occurred on the maternal allele.
Meaning the copy of the gene inherited from the mother.
Exactly. The researchers hypothesize that this might be due to something called negative selection against these particular variants in the male reproductive cells, the sperm.
So maybe it's harder for sperm carrying this variant to function properly?
That's the idea, though the precise biological mechanisms behind this are still being investigated. This kind of parental origin bias isn't always seen in genetic disorders, making it a potentially unique avenue for further study.
So digging deeper into this RNU4-2 story, it sounds like the researchers could even link variants in very specific regions of the U4 snRNA to how severe the neurodevelopmental issues were.
That's precisely what they found. It's quite remarkable. Variants located in a region of U4 called the T loop, which includes that N.6465 NST insertion we just discussed,
Right.
were generally linked to more severe neurodevelopmental disorders.
More severe how? What did that look like?
Well, these more severe cases often showed signs even before birth, with conditions like abnormalities in brain structures, such as the corpus callosum, which connects the two halves of the brain, and enlarged ventricles. Those are the fluid-filled spaces in the brain, as well as intrauterine growth restriction, meaning the baby didn't grow as expected during pregnancy.
Wow.
After birth, these individuals commonly experience significant developmental delays, severe intellectual disability, often being nonverbal, low muscle tone or hypotonia, and difficulties with feeding.
That paints a clear picture of the significant challenges associated with these two-loop variants. What about changes in other parts of the U4 molecule? Were the outcomes different?
Yes, they were, which again highlights this structure-function relationship.
Yeah.
The study indicated that variants in other regions of U4, specifically areas called STEM-I and STEM-3,
Okay.
like specific changes called N.76CT and N.7273del, tended to result in milder phenotypes.
Milder meaning.
Generally less severe developmental delay and intellectual disability, and some even retained the ability to use language to some extent. Notably, all patients identified with variants just in STEM3 were able to walk and speak to varying degrees, and their intellectual disability was classified as mild to moderate.
So there's a real spectrum depending on where exactly that U4 molecule is altered.
Exactly, a spectrum linked to specific functional domains within this tiny RNA.
So how do these alterations in the U4 snRNA actually interfere with the splicing process? What's the knock-on effect at the molecular level?
That's the key question. The study indicates that RNU4-2 variants can disrupt the crucial interaction between U4 and another snRNA called U6.
Ah, the one U4 keeps in check.
Right. This U4-U6 interaction is absolutely essential for activating the spliceosome and ensuring that the splicing machinery correctly identifies the 5' splice site that's one of the precise locations where the introns need to be cut. Certain regions of U4, like the quasi-pseudoknot and in STEM3 we just mentioned, are thought to be important for correctly positioning a key part of U6 that's involved in this recognition.
Okay.
So if U4 is altered, especially in these critical regions,
It can't interact properly with U6.
precisely, and that disrupts the whole activation process. In the more severe cases, the researchers observed increased usage of alternative 5' splice sites.
Meaning it cuts in the wrong places sometimes.
Exactly. It's just a loss of precision, a loss of specificity in the splicing process. It's like the editing tools are no longer cutting exactly at the intended locations, leading to errors in the final genetic message, the mRNA.
Which would then lead to faulty proteins or the wrong amount of protein.
Exactly. And that can have major consequences, especially during brain development.
And the research didn't stop there, did it? The study also looked at something called DNA methylation patterns. What did they discover there?
This was another fascinating layer to the findings. The researchers identified distinct DNA methylation signatures associated with RNU4-2 variants.
Okay, remind us what DNA methylation is.
Sure. Think of DNA methylation as adding tiny chemical tags, like sticky notes, to the DNA itself. These tags don't change the underlying genetic code, the A's, T's, C's, and G's.
Right.
But they can influence how active genes are. They can act like switches, turning genes on or off, or maybe dialing their activity up or down. It's an epigenetic mechanism.
So it's like another layer of control on top of the gene sequence itself.
Exactly. And the fact that these RNU4-2 variants are linked to specific, unique methylation patterns further strengthens the idea that they play a significant role in neurodevelopmental disorders. It suggests they can have broader effects on how genes are expressed in the brain beyond just the splicing errors.
Okay, so that's a really deep dive into RNU4-2, but the study also highlighted another snRNA, U5, encoded by the gene RNU5B-1, didn't it?
Yes, absolutely. Building on these compelling findings for RNU4-2, the study also uncovered significant insights into the role of another crucial snRNA, U5, encoded by the RNU5B-1 gene.
Okay, what did they find there?
Well, they found a significantly increased number of de novo variants in RNU5B-1 in patients with these neurodevelopmental disorders compared to individuals without them. This strongly suggests that RNU5B-1 is a newly implicated gene in these conditions, another piece of the puzzle.
And just like with RNU4-2, were there specific regions within the U5 snRNA that seemed particularly sort of vulnerable to these genetic changes?
Absolutely, a similar pattern emerged. The researchers highlighted a clustering of variants within a highly conserved region of U5, specifically a part called the 5-foot loop I.
Loop I, and what does that part do?
Well, this region is thought to be critical for U5's function in correctly aligning the exons at the splice sites during the splicing process.
So holding the pieces together that need to be joined?
Essentially, yes, making sure the right pieces of the genetic message are joined together accurately after the introns are removed.
Were there any specific recurring variants in RNU5B1 that the researchers identified, like the extra T in RNU4-2?
Yes, there were several recurrent variants found within this critical 5-foot loop I region. They mentioned specific changes like N.39CG, N.4243NTSA, and N.44AG.
Okay.
And importantly, these specific genetic changes were not found in large databases of genetic information from control individuals from people without these disorders.
Which strengthens the case that they're actually causing problems.
Exactly. It supports the idea that they are likely to be pathogenic or disease-causing.
And how might these RNU5B1 variants be impacting that crucial process of splicing?
Well, given that the U5 loop I directly interacts with the exons at the splice sites, essentially holding them in place so they can be joined, variants in this region could certainly affect the accuracy of splicing. The study's findings suggest that the impact might be specific to each variant, potentially affecting how both the 5- and 3-foot splice sites, the beginning and end points of the introns, are used.
So, again, a loss of precision.
A loss of precision, potentially in a different way than the U4 variants. This might also help explain the clinical variability observed in patients with RNU5B1 variants.
Variability like what?
Well, the study mentioned observing a range of features and even included seemingly opposite effects on head growth, some with small heads, some with large heads in different individuals with variants in this gene, so it's complex.
Interesting. The study also briefly mentioned another gene for U5 snRNA, RNU5A-1, didn't it? Was that significant too?
Yes, they did mention it. While the findings weren't as strong or as numerous as with RNU4-2 and RNU5B1, the researchers did observe de novo variants in RNU5A1 in a small number of patients.
Okay.
These patients also had neurodevelopmental disorders, along with a variety of congenital malformations, meaning physical differences present at birth. So this suggests RNU5A1 might also play a role in some cases, but more research is definitely needed to clarify its contribution compared to RNU5B1.
Okay. So stepping back now to look at the bigger picture, what are the major implications of all these findings? What does this tell us more broadly about neurodevelopmental disorders?
I think connecting this to the bigger picture, this research really underscores the significant role that new de novo variants in snRNA genes, particularly RNU4-2, and now it seems RNU5B1, play in the genetic basis of many neurodevelopmental disorders.
Disorders that were previously unexplained, maybe.
Exactly. Cases where standard genetic testing didn't find an answer. It highlights just how incredibly important the precise structure and function of these U4 and U5 snRNAs are for ensuring the fidelity, the accuracy of the splicing process.
And splicing is just fundamental, right? Affecting tons of genes.
Absolutely fundamental. It's essential for the correct expression of a vast number of genes, especially those critical for the developing brain. So Errors there can have widespread consequences.
And it's interesting to consider that this study focused on a specific set, 50 well-known snRNA genes. Does that mean there could be even more to this story, perhaps involving other types of small RNAs or related sequences?
Absolutely. That's a great point. The researchers themselves point out that while they focus on these 50 well-characterized snRNA genes, our genome contains many more sequences that look similar. These are often annotated as pseudogenes, basically. They look like genes, but were historically thought to be non-functional evolutionary relics.
Okay, junk DNA again.
Kind of, but that view is changing. A very recent finding, actually, about one snRNA pseudogene called RNU5B-1 suggests it might actually have a function and be linked to a neurodevelopmental disorder.
Really? A pseudogene doing something important?
It seems so. This opens up the really exciting possibility that there are other seemingly non-coding RNAs out there, maybe even some of these pseudogenes, that could also play roles in genetic disorders. It adds another fascinating layer to our understanding of human biology and gene regulation.
Wow. So to kind of bring it all together, what are the key takeaways for us from this deep dive into snRNAs and neurodevelopment?
Okay, I'd say the main points are: 1, variations in the genes for RNU4-2 and RNU5B-1 are now strongly linked to neurodevelopmental disorders. These aren't minor players.
Right.
2, specific variants and their locations within these tiny RNA molecules often correlate with distinct clinical features. The genotype-phenotype correlation is quite strong.
Location matters.
Location definitely matters. 3, the fact that RNU4-2 variants, particularly that one specific insertion, seem to arise predominantly from the mother is an intriguing observation needing more study.
Mm-hmm.
And overall, this research just emphasizes the incredible precision required in the fundamental cellular process of splicing, and how even subtle disruptions in the machinery, these tiny snRNAs, can have significant consequences for healthy brain development.
It truly is an aha moment, isn't it, to realize that these incredibly complex processes are happening constantly at a molecular level,
All the time.
and that tiny changes in these minuscule molecules can have such profound effects on someone's life. It really makes you appreciate the intricate balance of it all.
It absolutely does. It highlights how interconnected and delicately balanced our biological systems are. And this research, it not only provides crucial insights into the genetic underpinnings of rare diseases, which hopefully offers hope for better diagnosis and understanding for families,
Yeah.
but it also deepens our fundamental appreciation for the intricate and often hidden processes that keep us healthy. For anyone fascinated by genetics, medicine, or simply, how the human body works, this study offers a really compelling look into a critical cellular mechanism.
Yeah, definitely. If you found this deep dive into snRNAs fascinating and want to explore further, there's some excellent resources available online. You could look into RNA biology, the Spliceosome itself, or the specific neurodevelopmental disorders we touched on, reputable scientific websites, maybe organizations dedicated to genetic research. Those are great places to start your exploration.
For sure. And this all leaves us with a really thought-provoking question, I think. Given that we're only just beginning to uncover the roles of so many non-coding RNAs, these snRNAs, and potentially others,
Right.
what other hidden layers of genetic regulation might we discover in the future? And how might these discoveries ultimately transform our understanding of health and disease, perhaps leading to entirely new ways to diagnose and maybe even treat these complex conditions down the line?
A whole world hidden in the junk? Fascinating stuff.